Welcome to The Explainer. Today, we're diving into a biological puzzle that's happening inside you right now, billions of times a second. We're talking about protein folding, this absolutely incredible process that takes a simple string of molecules and turns it into a complex 3D machine that's essential for, well, everything in life. So the question is, how does nature pull off this microscopic origami so perfectly and so unbelievably fast?
So let's just jump right into the big question, the one that has fascinated scientists for decades. How on earth does this floppy, one-dimensional chain of amino acids just spontaneously fold itself into this incredibly precise, functional shape? I mean, think about it. It's like throwing a piece of string in the air and having it land as a perfectly tied, intricate knot, all on its own.
And this slide right here, this just perfectly captures the challenge we're talking about. On the left, you've got a random, tangled mess. That's the unfolded protein chain. But on the right, you have the protein myoglobin, this beautifully structured and totally functional molecule. It's just a jaw-dropping transformation from complete chaos to perfect order.
All right, so let's start by just wrapping our heads around the sheer scale of this puzzle. You see, a single protein chain could theoretically twist and turn itself into a mind-boggling number of different shapes. So the real mystery is, why does it always, consistently, pick just one specific, functional shape out of all those endless possibilities? You know, even the scientists who first laid eyes on a protein structure were just blown away. When John Kendrew first saw the structure of myoglobin back in 1958, he wasn't just impressed, he was completely baffled. He was expecting to find some kind of simple repeating pattern, but what he found instead was a level of complexity that, frankly, no theory at the time could even begin to explain. Okay, so to really talk about this, we need a key term, conformation. All this means is any of the possible 3D shapes a protein can get into just by twisting and rotating its chemical bonds. Now here's the wild part. For a typical protein, the number of possible conformations isn't just big, it is astronomically huge. We're talking more than the number of atoms in the entire universe. And yet, out of all those countless astronomical possibilities, the protein somehow, every single time, finds its way to what we call its native state. You can think of it as the protein's home. It's that one single functional folded shape where it's the most stable and comfortable. It's a state of lowest energy, perfectly designed for the job it has to do inside a cell.
So what's the secret sauce holding this native shape together? Well, it's not one or two super strong bonds. Instead, it's the combined effect of thousands of tiny weak ones. And the biggest driver of all is something called the hydrophobic effect. Imagine shaking a bottle of oil in water, you know? The oil droplets all clump together to hide from the water. That is exactly what happens inside a protein. The oily, water-hating parts of the chain desperately try to get away from the water around them, so they bury themselves in the protein's core. This powerful inward pull is what literally scrunches the whole thing into its compact, stable shape.
Okay, so we know that protein has to fold up, but how does it even start? Let's look at the very first steps in this origami puzzle, the basic folding rules that create these regular repeating patterns. These are like the fundamental building blocks of all protein architecture.
The first and most common of these patterns is the alpha helix. What happens here is the protein's backbone twists into this tight right-handed coil. It looks a lot like a spiral staircase. And you can see how the side chains, the parts that make each amino acid different, stick out from that central column.
Now, how does this helix stay so perfectly in place? Well, it's held together by this beautiful repeating pattern of hydrogen bonds. If you look closely here, you can see them. These weak bonds act like tiny little staples, connecting each turn of the helix to the one just below it. And that's what makes the alpha helix such a stable and rigid piece of the final structure.
All right, the second major pattern is the beta sheet. In this case, instead of coiling up, different parts of the protein chain stretch out and line up right next to each other. This creates this kind of pleated, sheet-like structure. It's also held together by hydrogen bonds, but this time the bonds form between the adjacent strands, locking them together side by side. But of course, you can't build a complex 3D shape with just straight rods and flat sheets, right? You need connectors. And that is where these really elegant little structures called beta turns come in. They are absolutely crucial. They let the protein chain make a sharp 180-degree U-turn, which allows it to fold back on itself and create that compact, globe-like shape.
So now we've got our basic building blocks, the helices, the sheets, and the turns. Let's see how they all come together to assemble the final functional 3D sculpture. And for that, we're going to go back to our friend myoglobin, because it is the perfect example of this tertiary structure.
And here it is again, myoglobin. Its overall 3D shape, what we call its tertiary structure, is built almost entirely from 8 of those alpha helical segments we just saw. You can see them here as these ribbons, all packed together in this very specific, very intricate arrangement, and they're all connected by those crucial bends and turns.
And if we switch over to this space-filling model, wow, you can really get a sense of just how incredibly dense this protein is. I mean, there's almost zero empty space inside, and that interior, that's the hydrophobic core we talked about earlier. All the water-hating bits are crammed in there, hiding from the watery surface, and that is the key to locking the whole structure into its stable, functional shape.
And this slide, this is what it's all about. This shows you the ultimate purpose of all this complex folding: function. See, the intricate 3D structure creates this perfect, protective little pocket for a special molecule called the heme group. That's the red part there. This is the exact spot where myoglobin grabs onto oxygen. Without this precise 3D shape, that pocket wouldn't exist and the protein would be totally useless.
So, we've built our molecular masterpiece, but here's the twist. The very same forces that create it, all those thousands of weak interactions, also make it incredibly fragile. So now let's take a look at how easily this intricate structure can be taken apart in a process called denaturation.
So what happens when that delicate balance of weak forces gets disrupted? Well, the protein loses its native 3D structure. That's denaturation. And because a protein's function depends 100% on its shape, when it denatures, it just stops working. The lock completely loses its keyhole.
The process itself is actually pretty straightforward. You start with the perfectly folded native protein. Then you hit it with some kind of stress, like heat. I mean, just think about what happens when you cook an egg, right? The heat energy is enough to break all those weak hydrogen bonds and mess up that hydrophobic core. And the result? A denatured protein. An unfolded, tangled up chain that has completely lost its function.
And this leaves us with a final, just staggering thought. That native folded state of a protein, it's only marginally stable. The tiniest shift in temperature or pH can be enough to make it completely unravel. So the real puzzle isn't just how a protein folds once, but how our cells manage to build, maintain, and protect trillions of these delicate, absolutely essential machines every single second of every day. It means the cell isn't just an architect, it's a constant, vigilant maintenance crew, working tirelessly to keep the entire machinery of life running.